Hello, everyone, and welcome to ACS webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I'm pleased to be your host for today's broadcast, which is being co-produced with the ACS Division of Chemical Toxicology, the ACS Division of Medicinal Chemistry, and the American Association of Pharmaceutical Scientists, and ACS Publications. Today, we will be joined by a team of chemists from Janssen's pharmaceutical companies of Johnson & Johnson. Yvonne Will, the Vice President of Predictive and Investigative Technology, and Kevin Coe, who is a Senior Principal Scientist. Yvonne and Kevin will be discussing the current understandings of the mechanisms that contribute to drug-induced liver injury and how advances in reactive metabolism trapping have improved detection capabilities. Our moderator for today is Kaushik Mitra, who is currently Director in the Department of Drug Metabolism and Pharmacokinetics at Janssen Research and Development, based out of Springhouse, Pennsylvania. In addition, he serves as the head of the Biotransformation Group and the DMPK lead of the Cardiovascular and Metabolic Disease Therapeutic Area, overseeing an active portfolio of multiple internal and external assets. And with that, I will turn it over to you, Kaushik, to get us started for today.
Thank you very much, Mike. That was a beautiful intro. Let's get this show on the road. Drug-induced liver injury, of course, is something that we are all familiar with in this industry of making drugs. It's an interesting adverse effect that many times show up very late in a drug development paradigm, and perhaps even sometimes well into the clinic, which makes the prediction of this for promising drugs quite challenging. But there are some approaches to try to mitigate that, and what we want to get started on today is two talks that will discuss two of the mechanisms that are involved in drug-induced liver injury. To start off this series of conferences, we have Dr. Kevin Coe from Janssen Biotransformation Group. Kevin received his PhD in MedChem from the University of Washington with Dr. Sid Nelson, where he studied the role of structural alerts and how they play a role in drug bioactivation and cytotoxicity. He also then, for the focus of his work, was a nitroaromatic drug futamide, and a very interesting molecule for many of us who study this area. And following his PhD, he went to Janssen Research and Development, where he has been since. He is currently the Senior Principal Scientist and heads the Biotransformation Group at La Jolla. During his 13 years at Janssen, he has served as the functional lead for drug biotransformation and mechanistic in vitro teams, as well as the DMPK project representative in both discovery and development programs. Author of 13 peer-reviewed publications and 2 book chapters, Kevin brings a wealth of knowledge in the area of biotransformation. So with further ado, Kevin.
Thank you very much for the introduction, Kaushik. And thank you all for joining today. So as mentioned, my name is Kevin Coe. I'm from Janssen Research and Development. I'm based here in San Diego. And the title of my presentation today is Reactive Metabolism Trapping Applied to Drug Discovery, Its Applications, and Present Challenges. So since I have the honor of introducing the subject matter on drug-induced liver injury, I'll provide a little bit, maybe about a 10-minute introduction to the subject matter, and then I'll focus my talk on reactive metabolism. We'll talk about the factors that influence its detection. We'll talk about some chemistry then. We'll talk about some case studies of where we work closely with the medicinal chemists to address reactivity in our discovery program, and then we'll talk about present challenges before concluding. So let's first start off with a case study here. We have a patient taking a new anti-diabetic agent to treat her type 2 diabetes. Now, under the first 3 months of therapy, there's no adverse effects. She takes the medication once a day. However, after 3 months, experiences fatigue for 2 weeks, visits her doctor, and 3 weeks later ends up in the hospital. And what is found is that she has edema, nausea, emesis, and metabolic acidosis. A liver biopsy is done, and it reveals that there's severe hepatic necrosis that requires a liver transplant. And this is kind of an illustration of how scary these drug reactions can be at the liver. Now, this given drug agent was troglitazone. It was a PPAR activator, the first in class that came on the market in 1997 to treat type 2 diabetes. Now, during the clinical stage, approximately 2,500 patients were dosed with troglitazone, and there was a mild rise in ALT in about 2% of the patients, leading rise to 2 patients with severe injury. However, it was considered safe enough to place onto the market to treat diabetes. And once on the market, the numbers really expand, right? After 3 years of being on the market, 2 million patients were prescribed triglyceride. And only with the numbers being as such did the latent observations of hepatotoxicity become more apparent with triglyceride, where we had 63 instances of acute liver failure associated with the drug use. And in March 2000, given these safety concerns, triglyceride was removed from the market. So let's talk more about drug-induced liver injury. So I want to get everybody on the same page here. So oftentimes with orally administered drugs is absorbed in the gut, goes to the portal vein, and the liver is the first major organ beyond the gut to receive a high volume of drug. And the liver has a role to process this drug material. It has a battery of drug metabolizing enzymes and transporters to help the elimination process of the drug, so it doesn't build up and accumulate. However, that also means the liver has a high load or burden of drug that it has to process. And that's why it is oftentimes a susceptible organ. And greater than 50% of acute liver failures are due to drug-induced means. Now, most of that is due to acetaminophen, and a drug that we'll talk about in a few slides. But if we exclude acetaminophen, about 14% are due to other medicinal agents. And they can have a severe consequence, up to a 10% mortality rate. As we saw in the triglyceride example, we can have evidence of liver transplant. And we know that DILI is a major reason for drug failure. So it could be in the preclinical space when we do our safety studies in rodents or higher species. It could be during our clinical trials or phase trials, or post-marketing, as we saw in the triglyceride example. Now, between 1975 and 2007, it was found that for drugs that were being developed, about a third of them were withdrawn for DILI concerns. And I'd like to think that since 2007, right, over the span of 13 years, right, we've come a ways, and we have. There's been some excellent publications on the subject matter. There's been scientific advances, but we still have, in the pharmaceutical industry, notable drug failures for very late-stage assets across multiple pharmaceutical companies. So there's still very much a need to learn further, right, to better predict DILI. So why is it so challenging to predict DILI? Well, as exhibited by the triglyceride case, it's very rare. These are idiosyncratic reactions. They occur in less than 1 out of 10,000 patients administered the drug. They require long latency periods, as we saw 3 months for that triglyceride case study. It doesn't have it's not anticipated based on the drug's known mechanism of action. And there's no apparent dose relationships. We're not quite sure when to anticipate the effects to occur. There's also not a common risk factor that as we take our new medicines to the clinic, that we should anticipate when these reactions might arise. Furthermore, compounding it, the preclinical species we use cannot well exhibit these idiosyncratic reactions. So all of this culminates into a very limited mechanistic understanding for how these drug-induced liver reactions occur. We know them to be very complex, and they have as much to do with the drug as they do to these susceptible individuals. Now, the liver does many functions. It's also susceptible to multiple routes of toxicity. Today, what I will focus on is reactive metabolism. Yvonne will feature mitochondrial toxicity. We can also have lysosomal trapping, transport inhibition, formation of reactive oxygen species. So in one organ, multiple reasons where we may elicit damage. And indeed, if we look back at triglyceride in the literature, we find multiple explanations for why this drug may have caused these severe hepatotoxicity. Right? We have evidence of reactive metabolism of triglyceride, evidence of transport inhibition in plated human hepatocytes, cytotoxicity in HepG2 cells of parent drug and metabolite, and evidence of mitochondrial disruption of triglyceride. So all of this, either in isolation or in concert, may lead rise to the liver injury in these susceptible individuals. And that's also the big layer to it as well to make them so rare, is there certain factors in this individual that make them susceptible. It could be nutritional status, age and gender, their ability to drug metabolize, either bioactivate or detoxify drugs, polymorphisms in those machinery, or in the immune system machinery as well. All might play a very major role here. And it's very difficult going to the clinic as we discover new medicines to anticipate who might be most susceptible to a potential idiosyncratic drug reaction. So a lot of times in the drug industry, during discovery, we focus on the drug risk factors as the most relevant ways to maybe minimize this potential late stage. So what this means is that there's multiple mechanisms that might lead rise to liver injury from a drug. So we have multiple assays, we have multiple endpoints. And so one challenge in the pharmaceutical industry is to figure out how to piece these puzzle pieces together to shape a strategy to assess risk. And so today I'm going to focus on one puzzle piece. I'm going to focus on reactive metabolism. So before I get into reactive metabolism, I first want to emphasize that generally, drug metabolism is a benign and beneficial process. It's meant to take very lipophilic drugs, metabolize them into more polar metabolites to facilitate their elimination from the body, to prevent drug accumulation. So let's take the example of acetaminophen here. At pharmacological doses, a very safe drug. Now, there are enzymes within the liver, phase 2 pathway, sulfation, glucuronidation, that occur at the phenolic group of acetaminophen. And these form the benign sulfate and glucuronide metabolites that are transported and eliminated from the body. Now, there's a very minor pathway through cytochrome P450s to form a reactive intermediate known as N-acetylparaquinonamine, featured down here. Now, the body has a defense for this. The liver has very high intracellular concentrations of a nucleophilic scavenger known as glutathione that traps soft electrophiles like NAPQI and preventing it from eliciting any damage. The problem is with acetaminophen that if one should take too much, sometimes accidentally, given the prevalence of acetaminophen in many different products, these phase 2 detoxification pathways become saturated. More drug flows through the cytochrome P450 pathway, and we have more formation of the reactive intermediate NAPQI. So much so that it actually depletes these high levels of intracellular glutathione. And once that occurs, NAPQI then begins to alkylate proteins of the liver. And this is what leads rise to the hepatic damage. Now, acetaminophen is an intrinsic hepatotoxin. At a common dose, or all of us will have the same liver injury. So it's not considered an idiosyncratic drug, but it teaches us much about these types of idiosyncratic drug hepatotoxicity. Now, taking drugs like triglyceride, it's known that they can be bioactivated by these cytochrome P450 enzymes, these oxidative enzymes that exist in the liver. They can form reactive intermediates. They can alkylate certain types of proteins. These drug protein conjugate, right, is now covalent bound and can be processed and viewed by the immune system as foreign. And this may lead rise to an immune response to the drug insult. And it also has much to do with patients' individual susceptibilities, making them a risk factor here. So in the course of drug discovery, there are many different assays we are running along the way to better characterize our medicines. And we might see consequences of reactive metabolites in these types of assays. It could be in mitogenicity studies in Ames or micronucleus. It could be time-dependent inhibition of our cytochrome P450s. It could be in our safety studies in preclinical models or in our cell-based assays. But we're always very much concerned about the potential for an idiosyncratic toxicity, something that we don't well predict during the course of our discovery endeavors, and only happens, as you see, very late in the development stage, or worse yet, post-marketing. So what we do in discovery is we often look for the formation of reactivity early in the paradigm, right? So we look in our early chemotypes to see if there's any evidence for reactivity. We often use human liver microsomes. Now, this is a subcellular isolate from liver cells, hepatocytes, that contain most of the drug metabolizing enzymes. Particularly, they contain the cytochrome P450s that oftentimes have an ability to form reactive intermediates that might lead rise to alkylation of proteins. So how we conduct these studies is we first incubate the drug in these liver microsomes. We add in a NADPH to allow for P450 activity and a trapping agent. Now, this trapping agent could be the nucleophilic scavenger, glutathione, which is very good at quenching soft electrophiles like mycoreceptors. It could be cyanide, which is a good trapper of hard electrophiles like imidine. And the intent of the trapping agent is it helps to stabilize that reactive intermediate to permit for its detection by mass spec. Now, once we can detect it by mass spec, we can also then fragment, utilizing mass spec, the trapped addict into pieces, right, to hopefully identify what substructure on the drug is associated with that covalently bound trapping agent. And in so doing, we can come up with hopefully a hypothesis of how did a cytochrome P450 form a reactive metabolite at this given substructure to lead to the trapping agent. And this is the hat trick we're trying to do in our discovery programs. With detection, localization, and mechanism, we place our chemists under very good position then to think about clever chemistry to disrupt this bioactivation. Now, we know these assays have a benefit to aid our prediction on DILI. A nice work that was done by GSK back in 2012 took 200 drugs, 100 that were DILI positive and 100 that were DILI negative. Now, first and foremost, what they found is that actually the best predictor of DILI was dose. Higher dose drugs were more likely to be DILI positive than low dose drugs, right? So another strategy to minimize DILI is to lower that dose, right? So the more potent compounds that we are making, the more optimized pharmacokinetics that have the proper half-life and clearance values, right, is a strategy, right, to help minimize DILI likelihood. But in addition, what they found is there was value in addition to the assay. High-dose drugs, right, greater than 100 mg, that also form glutathione addicts in human liver microsomes, right, the evidence of reactivity, right, were 80% likely to be DILI positive from their analysis. So the combination of high dose and the evidence of reactivity is a bad combination, right? It's the main message. So now we think back to this scheme here where reactive metabolites might have consequences down the road. So the thinking goes, well, if we can work early in the discovery space, block reactivity, we might then have the ability to block the downstream consequences, right? Be they in discovery phase or later. So let's talk about some of the factors that influence an addict detection. That includes the test system that we'll talk about more on the microsomes, the MS instrumentation, and the software. And ultimately, these three things, system, the instrumentation, the software, makes us what I call the addict hunter, okay? A sea of spectra looking for these addicts, indicators of reactive intermediate formation. So first, let's start with the mass spec instrumentation. So over the last 15, 20 years, a various number of modifications have been made to improve our detection capabilities of reactive intermediates. They take glutathione analogs in certain ways offering benefits. So for first example, here's the ethyl ester glutathione, which enhances MS detection over glutathione itself by tenfold, enhancing our ability to find these addicts. Another clever trick is utilizing stable label glutathione at the nitrogens and carbons. And what we do in these is we incubate cold and a certain ratio of the stable label together into these incubations where we produce addicts now having a certain isotopic pattern. This allows us to then fish them out for the spectral information and better interrogate. It also allows us an ability to have an unbiased approach in trying to find certain types of addicts. Other modifications include quaternary ammonium GSH, and the one below is danzyl GSH, which allows for fluorescent detection. This allows for more semi-quantification to be possible for how much addicts are produced. So let's now talk about some of the more powerful instrumentation that is available nowadays. So the systems have gotten faster, right? And that means in this high resolution era, we're able to do more with a single injection. Take this example here, where we can do in-source fragmentation to produce a 272, a diagnostic product ion of glutathione additives. We can also, in the same scan, do negative mode scanning, full scan and data dependent, to see what ion led rise to 272. That's the addict we need to figure out. And we can also perform a positive mode, full scan and data dependent analysis to get the rich spectral information oftentimes that allows us to do that localization endeavor, right? That ultimately leads rise to the final point of the mechanism. All right. So with more powerful software, or more powerful instrumentation, this also means we need software to keep up. So this is an example of a software often utilized called Mass Metasite, which allows us to take the spectra we get from these instruments and look for and fish out these addicts, right? And this can be done through multiple orthogonal pathways, one of which is looking at the metabolite structure and looking for the formation of certain types of masses that are indicators of glutathione addicts. Also, the software can look in the MS-MS spectrum, look for certain types of neutral losses that are indicators of glutathione addicts, commonly neutral losses of 129. Certain product ions like 308, which is the intact glutathione, might also be a diagnostic feature for an addict. And if we do those stable label incubations with cold and stable label GSH, we have an isotopic pattern, which allows the software to fish out those types of addicts. So you can see multiple ways in which the software is able to help the analyst identify these addicts in this ocean of spectra. Now, one of the challenges when we come to test systems is that more often than not, chemists are very good at making metabolically stable molecules, right, that exhibit very low turnover in our in vitro assays. So here's an example of one program, very stable, very little turnover in human liver microsomes. And what we see is that that does not necessarily mean one cannot find an addict. We do. For multiple compounds, we actually see, despite the low turnover, evidence of that glutathione addict, right, indicator of reactivity. The concern is a compound like number 8 here, right, where there's no evidence of phase 1 metabolites being produced, and we don't see a glutathione addict. Well, does that mean that it's negative in a trapping assay, or does it mean that we didn't have sufficient metabolism to really appreciate its reactivity potential? And that means that addicts may go missed early in discovery and carry on later down the pipeline. And that's the concern. And we have examples like this. We had one particular molecule that was negative, low turnover drug, negative turnover and trapping, was not found to have thiol addicts in a suspension human hepatocyte assay. But later, when conducted in vivo metabolite investigations in the bile or rat, it was a major pathway to form thiol conjugates, an indicator of reactive metabolism. And in turn, when we took this molecule and used more powerful metabolic systems, these co-culture models I'm going to introduce soon, we saw that in human systems, we're able to produce the very same thiol addicts, right? So it's a challenge to capture these low turnover compounds early, utilizing the conventional microsomal conditions. So we want to get a better beat on this earlier in discovery, right, to help prevent that from cascading further. So we took microsomes, right, which is often a very convenient, cost-effective, and throughput means of doing these assays, and try and see if we can improve conditions slightly to address these lower turnover drugs. And there's a few factors that we looked at. We took a panel of known commercial drugs that form glutathione addicts. We looked at certain variables from test concentration, trapping agent concentration, time, and the amount of protein. And what we found is we can make some modest improvements in the detection of glutathione addicts, right? With a higher test concentration, we're now more Vmax, pushing the formation of a metabolite in the incubation, particularly for those low turnover drugs. Boosting to 5 millimolar GSH, which is the most physiological relevant, actually for certain addicts can have a benefit in detection. And with slightly longer incubations, up to 90 minutes from 60, we can see slight rises with an addict. But again, it's modest, maybe a 3- to 4-fold rise in the detection of addicts. Not much benefit increasing microsomal protein, by the way. More binding occurs, so not much of benefit. Now, in addition, we also wanted to focus on our co-culture models, right? And L-Pharma has an example like this, a very low turnover drug. You utilize your microsomes, your suspension hepatocytes, and you find the first 2 chromatograms, very low evidence of any metabolism happening in mostly parent drugs. However, in the co-culture model, which are plated systems with hepatocytes and fibroblasts to help maintain metabolic capacity for multiple days, we're now awash in metabolites, right? So it makes us better positioned to anticipate what metabolically will happen to these drugs once they go in vivo. Because I want to add for low turnover drugs, although they may be slow in vitro, their metabolism will be complete in vivo oftentimes. So it is a risk. So we took that one chemotype that advanced further down the portfolio that was missed, and we took 6 analogs from it and introduced it into these enhanced liver microsomal conditions and into a human co-culture model. We also utilized rat liver microsomes because we knew that the bioactivation pathway was more pronounced in rodents than it was in humans. And what we found for these 6 analogs is all of them form a glutathione addict in rat liver microsomes. Now, the challenge here is that, well, that's a rat. So we wanted to have more human precedent. So in these enhanced conditions, relative to what we saw in our standard conditions, we actually saw an enhancement in the detection of addicts. Right? 5 of the 6 compounds now had detection of glutathione addicts in these enhanced microsomal conditions. Right? Now, in the co-culture models, it's important to emphasize you have to dig a little bit deeper, right? Because glutathione addicts are not a stopping point, right? They're peptidases within the liver cell that further process a glutathione addict into cystine glycine, cysteines, and then N-acetyltransferases will conjugate to form an N-acetyl conjugate. And these are the types of species that you need to be looking at in these co-culture models. And indeed, there's a nice concordance between what we saw in the enhanced microsomal condition to what we saw in the co-culture model, right? And generally, there's a nice alignment there, right? That we're a little bit more well-positioned for low turnover drugs. But I will emphasize, you may need to use co-culture models at some stage to better appreciate the full metabolic capacity of these types of molecules. So now I've showed you instrumentation and software and the test systems that we have to date. So we've kind of moved from the addict hunter. Now we have to move to the addict doctor, right? We have to fix the issue. And it's not just the biotransformation scientist that's involved. You need to closely partner with your medicinal chemist, and it's a team effort to fix this issue. So let's talk with a case study here, right? So this is number one here. We have an aminopyrimidine. And we see with this particular substructure, there are multiple ways from the incubation where cytochrome P450 will form a reactive intermediate, right? We can have an oxidation occur at the para position. Another round of cytochrome P450 metabolism will make a paraquinone-amine-like metabolite capable of reacting to that nucleophile glutathione. We also can have epoxidation occur at that pyrimidine ring, right? Also a good electrophile for glutathione to trap. And finally, down below, we can have oxidation occur at the methylene functionality. We can lose that as water to produce an imine and yet another soft electrophile for glutathione trapping. So what we can see in a very narrow space, right, we have three distinct mechanisms of bioactivation, right? And that's important for the chemist to know how complicated it is, because this will influence what actually ends up being the final solution. So when we started, we had some partial successes, right? The chemistry moved from amino linkage to now an O linkage. This blocks the amine. However, having the pyrimidine unsubstituted still meant we had epoxidations and additional oxidations that led to bioactivation at the pyrimidine. Now, instead, if we block and introduce a trifluoromethyl at the pyrimidine, we can block bioactivation at the pyrimidine, but we still have the amine functionality, right? Or still have the amine group to allow for amine generation of additives. So one full successful strategy was to lock in that oxygen and introduce substituents that allowed for metabolism elsewhere, pushing it away from the pyrimidine. So they introduced a methyl methoxy or a fused ring in the tetrahydroparan. P450s worked off these substituents into benign metabolites, and we did not have glutathione addicts resulting. Another solution is to completely block the metabolism, lock in the oxygen, and add groups like chloro and a difluoromethyl that block metabolism at that pyrimidine, also compounds that did not form glutathione addicts. Now you think, all right, we're good, we're fixed. Hold on, right? Discovery continues to optimize, and subtle changes on the molecule may bring back in reactivity. So the team introduced cyclopropyl at that methylene group, and we returned to having addicts in the very same area. In addition, we also might be optimizing other parts of the molecule. And the team's efforts to introduce a fluorophenol introduced a new site of bioactivation. So what this shows you is you have to remain constantly vigilant. It's never past you, that you always have to spot check to make sure you maintain success as we continue these optimization endeavors. Now in case study 2, we had a molecule with core structure and 3 distinct alkyl groups. We had addicts that were produced in liver microsomes that required cytochrome P450 activity. Unfortunately, the MS was limited. It couldn't really tell us where the addict occurred. All we knew is that it occurred at an aliphatic site, but we had 3 distinct aliphatic sites. So we needed to dig deeper into the spectral information. This is where you start looking at, well, what is my major metabolic pathway? So when we look at one example of such a molecule, we had metabolism really happen at 2 of these alkyl groups, at the left and right-hand side, leading to N-dealkylation pathways. Now, when molecules were predominantly N-dealkylated on the right-hand side through pathway B, these molecules tended to be clean in the glutathione trapping assay. Conversely, if teams found a way to block metabolism at the right-hand side of the molecule, preventing N-dealkylation through the introduction of a tert-butyl group, these molecules tended to form glutathione addicts. So we have a clue here, right? Now, what is it about this N-dealkylation pathway A that may be leading rise to a reactive intermediate? So we have to think about, how does this reaction occur, all right? Now for ND-alkylation, P450s oxidize alpha to that amino functionality. They produce an alcohol metabolite. Now this is a carbanolamine, tends not to be stable. It collapses into an aldehyde and free amine, and we detect the carboxamide as the metabolite in this instance. But this intermediate metabolite might have another route. We might lose water to produce an imine. And alpha to a carbonyl, we have a Michael-like acceptor that perhaps serves as a good site for glutathione trapping, right? And note in the process that we lose one proton from the drug molecule, all right? That'll be important for my next slide. So this is a plausible hypothesis, but we haven't proved it yet. So we worked back with the chemists, and they had a clever idea. Well, you know, one thing that we could do is we could introduce deuteriums in that methylene site, right? Now, if we don't find glutathione addicts, maybe we had a kinetic isotope effect, right, that would support the mechanism. If we find a glutathione addict that lost one deuterium, that also supports the mechanism. And if we find glutathione addicts that retain both deuteriums, that then suggests it's somewhere else on the molecule. Go fish, come up with a new mechanism. And what we found when we incubated this deuterated compound is that we formed a glutathione addict that had the loss of that one deuterium. And this helped prove the mechanism. Now this is important because once we achieve this stage here, teams are focused. They know exactly what substructure to spend time and energy on. And they have a mechanism that they can work towards to help decide how to block or disrupt this reactivity. So you can work very quickly once you know this. So the teams, first and obvious, they blocked it completely. They've introduced gem dimethyls at that methylene carbon, prevented glutathione formation. They also found that they could introduce sterics in this region, a single methyl at the methylene, or bochial groups adjacent to the methylene were also successful at blocking reactivity. In addition, they found that ring strain was beneficial. They introduced ring strain. This prevented bioactivation. If we have limited ring strain, this allowed for addicts to come back into play. And this is important. Now we can do very rational drug design, hypothesis testing to determine what is possible and what is not, to give ourselves options, right, as we continue to improve other properties on the molecule. Now, in case study 3, we had a program that had a significant aldehyde oxidase issue. It's another enzyme, phase 1 enzyme, that is found in the cytosol of hepatocytes and can metabolize heterocycles. And in this instance, it metabolized the pyrimidine group. We knew exactly where the site of metabolism was for AO. And what the team found a way to do is take that methyl ketone, tie that back into a 6-membered lactam, and block the AO metabolism. The problem being is we lost some potency in the molecule. But the team, undaunted, was able to find a way to bring back potency by contracting the ring by 1 carbon to make these 5-membered rings. We got back potency. However, we introduced a new problem. We brought in glutathione reactivity. So what's happening here? Well, in these 5-member rings, for those that are savvy in P450, one great site of metabolism now is that benzylic carbon. And that's indeed likely where metabolism happens. Oxidation happened there. And we also had a system primed for conjugation. We readily lose water. And much like case study 2, we form a Michael-like acceptor, right, capable of being trapped by glutathione. So now that we had an idea of likely how this was occurring, what are different solutions? Well, the team tried sterics. They introduced a single methyl group, or they tried positional isomers, moving carbons and nitrogens around, right, away from that benzylic orientation. However, these did not work. A single methyl, right, which worked in case study 2, does not work here. We still have an available proton for metabolism. The benzylic site is still a very preferred site, and we still pick up the same type of reactive Michael-like acceptor. Now, with a positional isomer, we might have changed the mechanism, but we still have the ability to make an amine metabolite that led rise, likely to its glutathione addict. So for these types of molecules, we had to have more aggressive blocking strategies. Utilize a gem dimethyl, which worked in case study 2, or exchange in case study 1, a nitrogen for an oxygen. In both instances, we still have P450 metabolism happening here, but we can no longer lose the water that led rise to the electrophile. Finally, in my case study 4, we have a triazole end linked to a pyrimidine. Now, in these incubations, we had no evidence for a glutathione addict in human or in other preclinical species. But when we took this compound in vivo, what we found in the rat bile was a prominent metabolite known as M9 that had the loss of this pyrimidine group, a dearylation event. And we had many downstream metabolites from M9, as you see in the mass spec chromatogram. So this was a very extensive pathway in rodents. And we never saw any sialax in the rodents. However, it was concerning, how does this dearylation pathway happen? Well, we got on a chalkboard and drew it out, right? Possibly we have an oxidation in a paraposition, another round of P450 metabolism. We have a paraquinonamine-like electrophile. Water can hydrolyze. We can produce the triazole in M9, and then we release a species that we would be concerned about, right? Something that looks like it could alkylate to protein. And we also know from the literature that drugs with a very similar type of dearylation mechanism, nifazodone, which has a phenyl tethered to a piperazine, can have a loss of this phenyl group through a very similar mechanism. And this nifazodone is a hepatotoxicant, so it's not a good association to have here. And what we found for this given drug here is that when taken into safety studies in rodents, it was hepatotoxic. It was an hepatotoxicant in rats. So what we did with the program is we worked on blocking this dearylation event from occurring. In subsequent analogs successfully doing that, these then were placed in safety studies and ended up being safe in the rodent, right? So there's some circumstantial evidence here that indeed this pathway was likely the culprit, even though we did not find an addict. So it's also a clue that we should also pay attention to unusual looking metabolites and do some further investigations, even if there's not the signature glutathione or cyanide addict. So let me focus now on some of the challenges that face us for trapping assays. So oftentimes I speak of the assay as a positive or a negative, a yes or no, right? As a very binary thing, right? We see an addict, we don't see an addict. But really, in reality, it has much to do with the instrumentation we're using. Eras ago, we had triple quadrupole mass spectrometers looking at neutral losses of 129 to detect glutathione addicts. But as I've shown you earlier in this presentation, the instrumentation has improved. They're more sensitive. We have better, clever ways to detect addicts. So our abilities are now able to detect lower and lower amounts of these glutathione addicts. And we'll continue to prove, right, as there's advancements in the software and the instrumentation to find lower and lower levels of addicts. So the assay, unfortunately, is not a risk assessment assay. It does not predict whether a compound will or will not be DILI positive. It is more of a hazard potential assay, right? So there are other key factors that make these addicts relevant or not. So this can hopefully be illustrated in my next example. So we take the three top drugs here, triglyceride that I featured earlier in the talk, clozapine, and nifazodone. All of these are DILI positive drugs. And it's known that they can be bioactivated by P450s to form reactive species. And we trap these addicts. We have glutathione and cyanide addicts for all three of the above, right? However, the analogs below, pioglitazone and rosiglitazone of triglyceride, olanzapine from clozapine, and buspirone from nifazodone, also form the very same addicts. However, these drugs are safe. They do not cause liver injury. So what's the big difference here, right? The top drugs that are DILI positive are high-dose drugs. You think back to that GSK paper, right? They're greater than 100 mg, right? The below-dose compounds that are safe at the liver, they're low-dose. They're less than 100 mg. So dose is also a critical determinant. And much like that acetaminophen example, right? The body might be able to tolerate a certain degree of reactivity, but overwhelming it might lead rise more to these types of toxicity. And the better detection is, the more likely we are to detect. We take our portfolio in La Jolla. If we run these trapping assays across 500 different molecules, we find that 70% of them form some form of ethyl of various abundance, right? And the challenge in drug discovery is we don't always know the dose. We don't know whether we're going to be 200 mg, 100 mg, or 20 mg in the early stages of discovery. So it's hard to discriminate how risky is this at an early stage, just based off a trapping assay, right? So work has been done over the last 10 years. A nice work from AstraZeneca, the Thompson paper, as we call it, helps to allow for more quantitation, right? Taking the observation of an addict to now how risky is it, measuring the amount of drug that's radiolabeled in these incubations, placed into human hepatocytes, to measure covalent binding to what we're concerned about, the protein. And then in addition, layering on the dose that we anticipate, right? So if we see a high level of covalent binding coupled with a high dose, right, we have more risk here. And they found that there was an empirical threshold, a 1 mg covalent body burden, right, that if drugs that were beneath that threshold were more likely to be DILI negative, and drugs that were beyond that threshold were more likely to be DILI positive. So this gives us a way, at least, to think about moving forward our drugs. If they have addicts, where do they fit on the body burden spectrum? Are they high or are they low? And this allows us to do risk assessment. And this is really where the science is moving to, is trying to better get quantitative, right? And also thinking back to those puzzle pieces to integrate this into a strategy. Take not just a trapping assay, but partner it with other types of assays and hopefully come up with a scoring system about how to progress things. And this is some nice work for Pfizer as well as AstraZeneca on the subject matter. Also, a nice recent work coming out of Merck that Koshik was part of, as well as others, helps to look at the consequences of reactive metabolism, looking more at the biological element of it, leading rise to activation of stress response signals, right? So a little bit more complicated matrix and more powerful system that now appreciates the biology behind a functional consequence. And that's more to follow there. So in conclusion of my talk, reactive metabolism is one of many risk factors that lead rise to drug-induced liver injury. Now the test models, the systems, they'll continue to advance. And preventing reactivity is challenging, as I featured in our case studies, but I want to emphasize is surmountable, right? On the level of the biotransformation side, it comes with trying to do the localization endeavors and come up with a plausible mechanism to feed back to our medicinal chemists, all right? And if they're committed, we come up with good hypotheses, we then are able to fix the issue, and it's surmountable, right? Now, where the science needs to go is detection is becoming easier and easier, right? The challenge becomes, what is the risk of that given glutathione or cyanide addict? And that's where the science needs to continue to evolve, is the biological consequence. And how do we learn that earlier rather than later? Now, the intent is that the more we address reactivity in discovery, it's less likelihood for concern deeper into development during our late stage clinical trials and post-marketing, right? To bring forward safe medication. So before I conclude, I'd like to acknowledge a few folks within Janssen Research and Development. Up top, in the time of COVID here, we have our Zoom meetings, and here is our global biotransformation core team. We have Phil Gherkin, Dave Evans, Koshik Mitra, H.K. Lim, and Laurent Leclerc. Down below is my addict hunters in La Jolla campus, Charlie, Wei Shen, Kai, Shu Yan, and Vishal. I'd also like to acknowledge the medicinal chemists at the La Jolla campus that we work very closely with to address this issue, to make that commitment to bring forward less reactive and hopefully very safe, effective medicine. And also my partner, such as Yvonne Will in toxicology, to help contextualize DILI in this reactivity in this broader ocean of DILI. So thank you for your attention, and now I shall hand over back to Koshik.
Fantastic, Kevin. That was a very nice summary of 20 years or plus years of work in 45 minutes around reactive metabolism. What I do want to take time, a couple of minutes to switch between Kevin and Yvonne, is to start off by calling out the Division of Chemical Toxicology within the American Chemical Society, who is the sponsor of this event, along with the ACS itself. I've been part of this division for more than 25 years from graduate school, and it's been a really great learning experience, where this is probably one of those rare organizations that brings chemistry, biology, biochemistry, metabolism, and toxicology on one platform. This is a rare combination to have, and I just wanted to give a shout out and thank you to the organization for letting us put this really important parameter in drug discovery to you all. Let me quickly take the opportunity to introduce Dr. Yvonne Will. We are really fortunate to have her here, finding time from a busy schedule to do this talk with us. She is currently the Vice President in the Department of Non-Clinical Safety at Janssen, and she leads a predictive, investigative, and translational toxicology group, where she leads a global international multidisciplinary team of fantastic scientists really putting together chemistry, biochemistry, toxicology, and helping make better drugs. Prior to Janssen, which she has been now about a year now, she spent 17 years at Pfizer, where she held multiple positions of increasing responsibility in the later years as the technology strategy and innovation lead. I know something that she is very passionate about. And the idea was to come up with new tools and strategies. And one of those tools and strategies around mitochondrial biology and toxicology is now adapted across the pharma industry as a platform for mitotox assessment. Dr. Will obtained her master's and PhD in biochemistry, biophysics from Oregon State University with Professor Donald Reed, studying the chemistry of drug-induced oxidative stress and its effect on GSH status in various organs, cells, and organelles. She's a prolific writer, having more than 75 publications, invited chapters, numerous internationally invited presentations, as well as national ones, and she has edited 4 books. It is my absolute honor to have her here today in the discussion. Yvonne?
Well, thank you very much for the kind introduction, and I don't think I deserve all that praise, because really everything that I show around the world, it's really been a team approach, and I was very fortunate to be surrounded by very talented and brilliant scientists. And interesting enough, as Kojik said, I've graduated into many different functions. And so this is all a long time ago, it seems, but it's still around. It's still coming back. I was just witnessing a program this morning. So we need to keep talking about it. We need to keep understanding it. And as Kevin nicely explained, we can develop a lot of tools, and we have done that very successfully. However, the translational aspects are still very, very difficult. So I looked at the clock, and I'm really glad that Kevin took all the time he did to explain, but I'll probably skip through a few slides and leave you with the handouts and the downloads. And please, email me anytime you have some questions, but I won't be able to walk through everything, because I know many of you on the line are really eager to ask some questions to Kevin and myself, and as well as Kaushik. So here's the outline for today, the business case. We already spoke about that mitochondrial function, dysfunctions. You show you some drugs and their targets, and then we'll talk about some assays that can detect it and the things we developed, as Kaushik said, to really deploy within industry in a high throughput format. I will talk just very briefly about the animal models and the dilemma we're facing. I think Kevin already mentioned it. Idiosyncrasy is not something that you will detect in the rat or in the dog. And talk a little bit about multifactorial toxicity, again, bringing together that puzzle, some novel biomarker approaches. So we do know that 40 drugs have been attrited, Kevin mentioned that, over the past years, but what he forgot to mention is that 13 of those were reported to actually exhibit mitochondrial toxicity. Nifazodone, benzerone, amyneptin, cetaxetine, triglydzone, tolcapone, fenformin, buformin, fluoxetine. So many of those are actually exhibiting death before they were attributed off the market. This is not my work, but the work of somebody who has made huge contributions to the field, really was trying to actually develop animal models I'll talk about later, unsuccessfully though. But again, had done an immense amount of work, really compiling the evidence that, mitochondrial liability could be detected for these compounds in various cell types, hepatocytes. And so here's a nice compile of literature that really built that case that mitochondrial toxicity is something we just cannot ignore. So everybody knows that mitochondria make ATP, but they also do a lot of other things. Steroid synthesis, fatty acid oxidation, heme synthesis, very important for calcium homeostasis. That's actually very much important for CNS functions, for example, the urea cycle, steroid synthesis. And they're also, the hallmark of apoptotic events. I don't know who's on the line, Kaushik could see it, but many people that have listened to my talks before know that I always get a kick out of this for several reasons. One is, yes, this is a mitochondria and not a pink movie. I will not ever make it in the arts, I guess. But this slide, I really call my job security slide. As I told you, I've worked many, many years with my team on this, and it is amazing how much there's still to learn about this little organelle every year. And we discover more and more its involvement in not just liver injury, but inflammation and CNS and aging, metabolic syndrome, you name it. So mitochondria really, a hallmark of many, many of those diseases, as well as organ toxicities. And the animation isn't working today, but what I wanted to share with you that when I started my career at Pfizer, as I said, there's many, many proteins, there's many targets in here, but what we did was we tried to build some assays around, some key mechanisms such as the inhibition of the electron transport chain, the uncoupling, where instead of ATP you make heat. That is actually, something they tried for weight loss programs. But again, due to safety reasons, as you all know, this had a bad end for some of the folks trying to use this mechanism to drop weight. Mitochondrial transition, pore opening, again, as a prelude to apoptotic events. And then within Pfizer, we had quite the program at that time in the UK for antivirals and antibiotics. And what we know, and I guess I should have told you this, is mitochondria actually have their own DNA. It encodes for some of the key enzymes in the electron transport chain, the catalytic units of these electron transport proteins, and also has a machinery for protein synthesis. And so antibiotics and antivirals, and I'll talk about this later, target those. And so we also ended up putting specific screening platforms around these type of programs. So what do these drugs do actually in mitochondria? And here's just really, a list. And again, you can look at it. There's ample evidence in the literature, but you see they can inhibit the fatty oxidation. They can induce mitochondrial permeability transition. They can work on the electron transport chain. And as I mentioned, antivirals and antibiotics particularly affect the mitochondrial DNA protein synthesis, but others do as well. And if you read across, for example, tamoxifen, I don't seem to have a pointer here, you see that it does fatty acid oxidation, that it has an effect on the electron transport chain, as well as on mitochondrial DNA. So we find out that many of these drugs are actually, we use the term promiscuous. They have many different off targets, and much of that is actually driven through the physical chemical property space. And so over the years, people have learned, chemists I should say, have learned that there is a close association between physchem properties, and even some structural alerts have been developed. So much has been learned, but certainly, not all of it today. Mitochondrial dysfunction in the liver can induce different types. There's microvesicular steatosis, and that can, under certain circumstances, depending on duration and severity, lead to profound hypoglycemia and encephalopathy. They can induce apoptosis and necrosis, so you will see cytolytic hepatitis that can again lead to liver failure. And mild mitochondrial inhibition will lead to macrovesicular steatosis, which under certain circumstances, of course, also can progress in fibrosis and cirrhosis. So interesting enough, and Kevin spoke about it, a lot of these reactions are idiosyncratic. And with respect to mitochondria, we do at least have hypothesis why that is so. Because what we know is that mitochondrial DNA has haplotypes. There are very ancient haplotypes. There are haplotypes specific to parts of the world. We know that certain diseases are associated with these haplotypes. There's, for example, an African haplotype L2, and there's a very close association to diabetes, for example. We also know that, for example, patients with particular mutations in mitochondrial DNA, once you put them on linezolid, they become deaf. We also know that folks with silent MELAS mutations under statin exposure can develop full disease. So the difficulty here is with the translation is that most of the time, again, looking at the drugs that we named and Kevin named, it's idiosyncratic. So often you have to expose many patients to get to really see the effect, and that leads to late stage attrition, and that is something that is not per se affordable from a patient care, but as well as also an economic perspective. So here are some drugs, again, just for you to look, that can cause the microvesicular steatosis. And you see this is not all one particular class of compounds. You see many different indications: antidepressants, NSAIDs, antibiotics, antidepressants, the whole spectrum. So just because you're working in a particular area that is not listed here, a therapeutic area, doesn't mean that you should not check for such liability. Here's a list of drugs capable of inducing microvascular steatosis and steatohepatitis. Again, what you see here is the variety of different drug classes that actually have exhibited this particular phenotype. So assays to detect mitochondrial dysfunction in vitro and ex vivo. We have made great progress. Technology has come a long ways. For example, when I was in graduate school, we had an oxygen Clark electrode, and that was about it. And as you know, a chemist wants to test many compounds, and they also want to generate full dose response curves. So this is where technology really advanced to provide that to industry. And you have several companies now that offer various assay kits and various platforms to do that. But when you have isolated mitochondria, which is still often the standard for this assessment, you can measure fatty acid oxidation. You can do this either with radio-labeled fatty acids, but you can also use respiratory screening technology. And what I have tried to do here is to give you a key reference you can go and read up or see how you might want to implement that in your research or in your company. Measurement of oxygen consumption with different substrates. That helps you to actually hone down on what is the site of inhibition. Is it a complex 1 inhibitor like fenformin or rotenone? Is it an ATPase inhibitor like amiodarone? So you can get more clarity, and that might actually help you with some molecular docking and maybe some computational approaches to try to modify your chemistry. You can also measure this through the respiratory chain complex activities. Again, these would be, isolating the enzymes or making the enzymes accessible and then doing some very targeted screening. And this is actually interesting because that's where we started out. We knew that there was many complex 1 inhibitors that had been reported in the literature out of academia for many years. And we thought, oh, if we can just screen that out, we would all be set. And then we found out, like I showed you on the very first slide today, that most of these compounds actually had multiple targets. And so we had to go back and say, well, we want a system more than just individual proteins. And that's really where the assessments in the mitochondria as well as the cells then came into play. Determination of mitochondrial transmembrane potential. Again, this can be done by electrodes, but also these days with flow cytometry and imaging. There's been, again, a lot of advancements in the technology to accurately monitor mitochondrial transmembrane potential, as well as the permeability transition pore. A lot of this can also be then detected in the whole cell. We do know that an organelle is just an organelle, right? It's a short-term experience. There's no P450. Drug has free access, so you could potentially either over-predict and under-predict. So people, have moved on to develop the same type of assessment really in cells. And what you see in the literature is mostly really have G2 cells and hepatocytes. And as Kevin alluded earlier today, that does not always really show you what the entire story is. He talked about slow metabolism. For example, again, when you think about mitochondrial DNA or protein synthesis, these are things, you know, that take time. So you need to have a system that has everything fully on board, transporters, P450, functional mitochondria. You need to have them living in your dish more than just short term if you wanted to prolong your exposure. And so what you see these days is a lot of talk about complex model systems such as hepatopac, liver chips, and a lot of investments have really been made in the last 5 years to understand the utility of these systems, also how to make them affordable, how to really get them into a high throughput format. So this is still an area of much research and ongoing technology development. But nevertheless, you can see here what you can potentially measure. You can stain for oil red for neutral lipids, lactic acid, ALT in the incubation media. Again, that is quite difficult and actually impossible for something like a HepG2 cell. However, in the hepatopac, you can get to that point where you can actually measure those biomarkers. You can, again, measure oxygen consumption through either static or soluble sensor technology. Certainly, the more simple models are amenable to high content imaging. And again, vendors are catching on really quick in that they're really trying to figure out how to you know, image also these hepatopac, liver chip, and other complex model systems here. There's one more, and I can't think of it right now, so forgive me, it will come to me. So good. Okay, so now it gets more complicated because you can do things in vivo, you can also do things in ex vivo. So ex vivo meaning you're either taking liver tissues or you're making mitochondria out of treated animals, right? And in vivo as well. And the difficulty here, I guess, to keep in mind is really this idiosyncratic incidents that Kevin talked about and I'm talking about here again. So this is known that these are not detectable in animals. All of this have been missed preclinically. That's how they got onto the market. You can detect mitochondrial dysfunction in animals. You can, but we do not know really properly how it does translate and from rats into humans. So still a lot of work to be done. Most of the translation has really been done from the in vitro straight to human, bypassing the animal. And I'll show you a couple of slides again to make my point of why that's been very difficult. But you have plasma biochemistry, lactate, ketone bodies, GLDH. There's even more than that. Urine biochemistry, as measured by mass spec, histopath. And then, as I said, you can do it, the investigations on the liver mitochondria or the hepatocytes isolated from these treated animals, as we just looked at it in the previously 2 slides. You can also take homogenates from these animals and look for, you know, again, activities, and you can subject them to PCR, to immunoblot analysis, whatever your question might be. So while you can do all of this, it is very difficult, though, to translate it. And again, any kind of you know, idiosyncratic type compound would not reveal itself in the animals. So why is that? So first of all, rats have a very, very high rate of drug biomarker formation elimination. Actually, their glutathione content is much higher, as well as their GST activity. They also have heteroplasmy, so they have a very rapid turnover of mitochondria. So you might cause damage, but they turn over very fast, and that will actually reverse any insult basically instantaneously. And they also have mitochondrial threshold effects that are quite different from human. So people have tried to do it. For example, there is a JVS mouse model that has a mitochondrial deficiency through changes in the carnitine transporter. And I will have to skip through this, but here's a successful example where they did a valproic acid treatment and actually could show at least apoptosis in the mice. Here's the SOD mouse model. I talked about Urs Burst-Turley. His lab studied this for years. And while they had success with flutamide in the sense that they could see hepatic necrosis, as well as also detect changes in enzyme activity indicative of mitochondrial dysfunction, they were unable to show this with other idiosyncratic compounds such as troglydizones, such as nefazodone. And so this is just one of these compounds that happen to work, but many of the others don't. So people have studied for years, tried to make model systems. There have been humanized model systems, and the story continues that if they tailor very much the experiment, that they can show it for maybe one of the drugs, but certainly not for the majority. And it's also very hard to do an experiment with the 200 drugs we spoke of that have these type of liabilities or have been treated or carry black box warnings in the clinic. So look through this yourself. I'm going to move on to just make sure that we have time still for questions later. So let me just show you a couple of the essays. You will have and had references all around here. So go look it up and see what maybe you're interested in or what might be helping your program. They all have their pros and cons. I think that's, again, another learnings we had in 10 years. No one essay will tell you the entire story. You have to tailor them. You have to understand what they can and can't do. And you have to be realistic. You have to communicate properly, to customers and chemists and teams of, really your recommendations and what you can really learn and translate from these essays. But this was a great success story with Laxell in Ireland, a great partner. They had developed soluble probes that could actually measure oxygen. So it was really nice in that when oxygen was depleted through mitochondrial respiration, the signal increased, and you could measure it very easily using just a plate reader. They were cell non-invasive, they weren't toxic, we could even multiplex them. So really great platform, and we did a lot of work for probably about 5-6 years to learn, how to run the platform, to adopt the platform to 96 and 384, to develop algorithms to read out the data in a proper way, as nanomoles per milligram oxygen consumption. So a lot of work was done, and I'm still incredibly thankful to my team that was involved in all of this. But here's an example of how this looks like. You can see in the left corner, it's an uncoupler. So as I told you, they don't make ATP, they respire and produce heat, and you can see a sharp increase in the signal. You have an inhibitor in the lower part of the left plate. So there's no oxygen consumption because it's all inhibited. And when you look at the left to the right side of the plate, you see there's a really nice replication showing the robustness of the platform. And so moving on from there, we wanted to know, can we actually truly run some dose response curves from the top to the bottom, different concentrations, and we won't go through this, but yes, the answer is yes, we can. But this is still a signal, right, of fluorescence, and we needed to learn how to now properly transform that into oxygen consumption. So 2 years later, we were able to do this, and here you see how this looks like. On the top is an NSAID and a mesylide, and you can see that different increasing concentrations of drug show the inhibition of the oxygen consumption. You see the same on the bottom for Sudexicum, but much less effect on that. And then again, talking about the limitations. So on the top here, you see Solandac sulfide, Solandac, and you see no effect, but we do know it's a hepatotoxicant. And so what you see here is when you actually use the reactive metabolite of Solandac sulfide, you now suddenly see the effect on the mitochondrial function. So the value here is that identifies inhibitors and uncouplers of the electron transport chain. It's very high throughput, it's reproducible, it's easy to use. You can rank order compounds within a series, and you can probably even use it to identify structure activity relationships. However, it can potentially overestimate since an isolated organelle is used. It's only acute effect. I should have told you that a run is about 20 minutes. You may need to actually pre-incubate mitochondria with the drug to see the effect. That would be the case for something like metformin or fenformin that work by the Nernst equation. So again, if you just run it in a standard mode, you might still be really missing several types of compounds that act through a somewhat specialized mechanism. And we talked about it, it does also not take into account the conversion of the parent drug. So while it is valuable, it will miss drugs. And so you have to think about how you would address this in a different way. And the way we thought about it was really doing this in cell systems. But there's an interesting story to this. So when we first looked into what people had done with mitochondrial toxicants in cells, and there was a publication by Peter O'Brien at that time. He used high content screening. We saw that most of these compounds we just spoke to you about didn't really have any particular toxic effects on the cell system you used, which was a HepG2. And I couldn't quite put my head around it because Peter was an exceptional scientist and I couldn't believe that he might have done something wrong. But I was working with a chap named Jim Dykens at that time, and he was kind of a walking encyclopedia. And he said to me, well, Yvonne, don't you know in 1929, and I was thinking, wow, that's a long time ago, Jim. He said, well, don't you know, people describe this effect of what's actually happening in these cells. We culture them in high glucose, and what they do is they simply don't use their mitochondria, and they will just really live of glycolysis. And most of you on the phone might actually know that that is then what you see, the lactate production in your tissue culture on a Sunday when it's all turned yellow. So, but anyway, we took this really to heart, and then we thought, well, then we must make them use their mitochondria, and you can actually do that. Because if you, instead of glucose, use galactose in your media, now suddenly, as you can see here in the case of the complex 1 inhibitor, rotenone, anti-myosin complex 3 oligomycin ATPase, you can see now that in the presence of these drugs in the galactose media, which is the black solid line, you now see that the cells become susceptible. And so what we learned from this exercise is that you had a ratio of the glucose to the galactose above twofold, that you could be quite sure that you were dealing with a mitochondrial toxicant. And furthermore, you could conclude from many, many studies we've done on many thousands compounds that in that case, when you got that ratio, that mitochondrial toxicity was most likely the driving mechanism of the overall toxicity observed. So this is a success story in the sense that actually within Europe, under the MIP-DILI consortium, under Horizon 2020, this work was continued, and companies and investigators were trying to repeat that and to expand on it to see if this was valid for them. And that's really what you want, right? When we developed the assay, we wanted to see, was that working for other industry? Did they agree with what we wanted to really put it into the implementation mode? So cross-validation is really the key here. And they did. They adapted it actually quite nicely. They modified it a little bit, but at the end of the day, they actually validated and published the paper with the recommendation, to use this really as the industry standard screening assay to detect mitochondrial function in prediction of liver injury. So, interesting enough, for the first 10 years, I did believe that the world evolved as only around mitochondria, because as you saw from our presentation publications, we could certainly at all times show the association to liver injury. We could rank order compounds. So again, like just Kevin said, we found that triglycerin was much worse than, for example, pioglitazone. My favorite compound, nefazodone, was way worse off than buspirone. And interesting enough, it wasn't until about maybe 5-6 years ago where we started to have crosstalk, because we're all experts in a particular field. So Kevin is a metabolite guy, And then there's folks that are transporter experts, and then we were mitochondrial experts. But we started to talk, right? And I said, well, what do you mean nifazodone inhibits transporter? What do you mean nifazodone has a metabolite issue? And so we learned quickly that drug-induced liver injury, as Karen started us off today, is multifactorial, and it's a puzzle that comes together. And so again, from where we were sitting, we looked at this, and again, we found, expanding on our work, that when you looked at mitochondrial function, the association there was very clear, and you can actually set a margin for the prediction of toxicity. And when you came in and you looked at the same time also at the inhibition of BSEP, you saw that the association again to severity of the outcome was right there. So in other words, compounds that were withdrawn from the clinic or had a black box warning, which you see on this left here, you see the black, that means that there were dual activity both in mitochondria and BSEP, versus if you look to the very right with compounds no mentioning, there was none. And so again, not to have time to talk about today, but there's a very strong association again with physchem property space and the promiscuity of these type of compounds. And then, as Kevin said, people have progressed, and you'll hear this in the webinar in a couple of weeks as we go on with the transporters. Everything else is when you put that puzzle together and you apply, computational tools to it. If you also look at your physchem, if you look at those, if you test enough, compounds that are positive or negative and very well annotated for clinical outcomes, you can actually start now moving finally from a hazard to a risk projection. Just one last about biomarkers. So the EMA is a little bit ahead of ourselves here in the States, in that they have recommended the determination of lactate as well as GLDH and OCT. So again, the difficulty would be if you would be going into phase 1, you would detect it and stop your drug. That is probably a good outcome. However, it could all be negative and you wouldn't see it in an idiosyncratic case until very, very late after launch. And that would be, really the difficulty here. GLDH and OCT are the mitochondrial enzymes, so this is much more specific than just measuring AST, ALT for liver injury in itself. Recently, microRNA has also been explored. Progress has been made. The last word has not been spoken for sure on that. And then people have tried to see what they can do non-invasive. Methionine breast test has actually applied to diagnosing of liver diseases, but certainly has not been really utilized to detect potential technique. And it's very hard to, do that in a proof of concept by, giving somebody a mitochondrial toxicant. That would be quite unethical. So the summary really is, mitochondrial toxicity contributes to DILI. I have to admit today it's a contributing factor. It's not the only factor. We can detect it using in vitro assays, but not just one assay will be enough to understand, all of the different nuances. Most pharmaceutical companies have implemented mitochondrial toxicity testing in the drug discovery process. I showed you the MIP-DILI work. Animals do not easily reveal it, and certainly not for idiosyncrasy. And non-invasive human biomarkers still needs to be developed and utilized in the clinic. Again, this would not apply to anything that would develop itself in an idiosyncratic nature. And again, some additional references for you. You also found them throughout the presentation, and I'll be happy to get on the phone or reply by email anytime you have some questions. Thank you very much.
All right, no, excellent, Javon. This is a really nice summary of mitochondrial tox, but I really appreciate you calling out the different contributing factors that come into play for DILI. And to that end, I'll start with you, Yvonne, with the questions that we have specifically for you. And then we'll go to Kevin, where there are actually quite a few very interesting questions there as well. So let's start with Yvonne. This is from Marcus, and this is a question I think many of us I've been curious about is at what stage of the discovery program or discovery trajectory would you recommend starting to look at mitochondrial dysfunctional assays?
I think everybody is in a different situation, but I think for large pharma, most people like to really do this in even already in series selection, where you might just take a handful of compounds from each series and you try to find out, you know, where you really sit with that series. Do you have an opportunity to do SAR? Do they all look the same, right? Do you have a variety of responses? And then as you move into head-to-lead development, that's where you want, you know, to make sure that whatever is going forward for candidate nomination, you know, has been tested. I think, you know, the most successful you're going to be if you select the best series and do it early on when you still have chemistry, right? It would be very detrimental at candidate nomination to suddenly realize that you have that liability and you have no backup. So that costs you probably about 2 to 3 years to start all over again.
And a follow-up to this question, Marcus, is if you knew you had a compound that had mitochondrial tox, would there be suggestions of SAR that you can point to lead the chemist into perhaps making a better molecule?
Yes, definitely. So a lot of work has actually been published. There's a paper by Russ Navin. There's also a couple of experts over in the UK. So structural alerts have been published. And of course, every company has their own experience and, you know, their own secrets. So, you know, that we probably won't want to disclose. But yes, there's some features, you know, that are known. Certain amides, carboxylic acids, fluorine decorations. So, but yeah, look into Russ Navin. And then there's a gentleman in the UK who will come to me. I will raise my hand when that name comes to me.
And the third question along the same lines is, if one does run this integrated mitochondrial risk assessment early in compound optimization, what panels do you recommend? What assays, in other words?
I guess I shouldn't take away from all the other webinars that you're still having, but, you know, right now we're working, most companies working on this, what we call a DILI risk matrix. And every company does their own, you know, little version of it. For mitotox, the front runners have been the glucose galactose because it can run in high throughput format. There are a lot of companies also run the isolated mitochondria, usually with the soluble Lexol sensors in high throughput. And then again, some companies also do some follow-up, more mechanistic type of assays, complex activities or seahorse. But I would say the high throughput screens that are deployed within pharma are the GluGal, as well as some isolated mitochondria and a high throughput oxygen consumption measurement.
No, thank you. Those are very, very clear responses, Yvonne. This is an interesting question to both of you before we transition to Kevin. This is more around legal liabilities of issues that we might identify in the clinic before, maybe prior to approval by the agencies or after. What is the legal liability of a company when a drug is withdrawn from the market because of DILI? Interesting question.
It is. It's a challenging one because, and then Yvonne and I are not lawyers, by the way. So
yes.
Yeah.
But there is. I mean, at the end of the day, you see it on the ads on TV, right? That we can be held accountable if we bring forward molecules that are not safe, and that's why there's so much focus on trying to partner together. We know DILI is complex. We know a lot of toxicities are complex, right? So we work together to hopefully bring forward medicinal agents that are very effective, but they're safe, right? Because at the end of the day, we also want to, you know, we're also very intent on making successful medicines, right? That we can be proud of, right? So that's why we take the commitment every day to make sure, you know, in my realm, right, that we have metabolism happen through benign pathways and that it's not a risky molecule headed for the clinic, right?
So to add to that, maybe we have made progress, right, with influencing the agency. So for example, the antivirals, that mechanism is really truly known as being an inhibitor of the gamma polymerase in the mitochondria. And so if you bring any antiviral forward, you have to prove that you do not have such liability. So there progress has been made. There was, and maybe Kaushik can speak best to this, there was a lot of push to do something similar for liver injury with respect to transporter inhibition. but that hasn't gotten beyond, I believe, a guidance document. But there's certainly, so that's again, you know, where industry likes to come together within consortia, like I said, when they revalidated the Glugal assay, because that's the only way when industry together, right, has a path forward of what they find, you know, unacceptable or what they find necessary. But Kaushik, I'll let you speak to it.
Overall, I think it's the idea of full disclosure and transparency. So everything that the pharmaceutical companies do is out there for agencies to review all the data. And if there is any indication of any kind of liability that starts to arise, there is 110% commitment to get ahead of it at any point of time, right? And actually being very, very super conservative on issues. So that's the reason why you always, many times you'll see these clinical holds put on programs, maybe at the slightest indication of risk. And then you would go back and try to figure out what happened. And the clinical trials will only start after we are convinced that the risk is not a general risk or cannot be it cannot be reversed. So, you know, there is obviously liabilities. And again, we're not lawyers. We cannot speak to the exact sort of the terminology or the extent of it. But from a science perspective, it's really it's about disclosure and doing your due diligence at any point of time.
All right, I think a lot of times we take very conservative assessments on things along the way, right? You know, that if our exposures are lower than anticipated. So we try and factor that in as we advance forward to the clinic.
And one last question from Nestor on the on what type of 14 carbon labeled fatty acids are administered for the whole body FAO experiments. This is the autoradiography experiments, the fatty acid oxidation experiments, I think what he's referring to. Is there a kind type that you can refer to?
Sorry, say this one more time.
Yeah, this is for when you're running a whole body fatty acid oxidation experiment. Right?
Well, I wouldn't, but
yeah, what kind of C14 compounds would you use?
You know, I'm not really familiar with it, and I can't say I've ever done it. I mean, we have done fatty acid oxidation as a target, right, and in mitochondria and the seahorse assays. So I have to pass on that. It was morely for completeness of, you know, methodology for this particular talk, but I've really never done it.
All right, so Kevin, let's move on to the biotransformation end of things, and there are actually quite a few there. Let me start with one question that sort of seems to be a trend here, a sort of underlying component. What is the correlation, and I'm smiling because we talk about this all the time, Kevin, what is the correlation of GSH adapt to protein adapts and to toxicology? So essentially, and then again, a related question, you can also bundle all of this together. Your sensitivity is so high in instrumentation that you'll probably always find something, right? And which is probably in the range of 70 to 80% of our compounds. How do you know whether a given amount of reactive metabolite will be relevant with respect to DILI? And from Adnan, a similar question on, is there data on what kind of percent of reactive metabolism contributes to DILI? And from Jorge on the correlation of GSH trapping positive and DILI. Is it possible the GSH trapping experiments are over-predicting? So you get the sense, right? It's a question of whether these GSH trapping studies are truly predictive of DILI, given the high sensitivity that leads us to find GSH adducts for almost all compounds.
I would say they're not predictive in the sense of a risk assessment, right? That as I kind of showed in that example with the analogs of triglycerone, pioglitazone, rosiglitazone, olanzapine, that you can find addicts in these assays, but the addict detection itself is not the decision maker on whether it will be DILI positive or negative. This is where the challenge is, I think, in the pharmaceutical industry is when you do these assays up front, right? More than likely you will find things, not always, but you will. And the question is when to react to it, right? And when is it acceptable, right? And when is this an acceptable molecule to move forward with this degree of an addict detected, and when to spend more time on it, right? So we have to move at some stage from this kind of hazard identification that we're doing to go, this is still a very optimized drug. It's got a lot of properties onto it. How extensive is this reactivity? And perhaps we move now to risk assessment. Now, I kind of illustrated briefly at the end that covalent binding study that was featured by the AstraZeneca Thompson paper. And that's a way where we move away from simply a detection, a yes or no, but rather how much and what are we binding to, right? And I think where the science needs to push is a little bit more is those consequences of, is there a consequence to the given addict that we have, right? So it's definitely a sensitive assay, but we also want to give ourselves the upfront bandwidth to fix things. Remember, discovery is the moment, right? Once you nominate that and put it in the clinic, that's the drug, okay? And so once that's there, we can talk about whether it's an issue or not, but we can't fix it then. So in the discovery space is the grand opportunity to improve the landscape of the chemistry and minimize the likelihood, and hopefully, make it a very competitive and successful molecule down the road. So it's just that moment of discovery when we go from hazard ID to risk assessment and doing additional assays to contextualize how risky is the assay, right? And that might be additional assays. We might partner with the Vaughn's group and look at mitochondria and additional assays. We might do covalent binding studies. We might try to appreciate how much flux of this pathway do we anticipate in humans. So it doesn't end simply with a trapping assay by any means, right? Compelling molecules should get through and be further characterized.
So Kevin, maybe to add to that, right,
Sure.
is that I think the one thing we all agree by now is that if a compound has multifactorial, so you can detect, everything, the cytotox, the mitotox, the transporter, the metabolite, we would be crazy to move these things forward any longer, right? I think we've gained enough experience from these retrospective analysis. And we also understand a lot of the physical property space. So I think the challenge will be that now we're actually starting to see, a compound, for example, that has only mitochondrial toxicity. And there's only two that I know in the entire world, right, that were like that. One was fenformin, and the other one is the compound that discontinued in Europe with Regan last year, right, due to acute death. But other than that, we don't know. So same for metabolite only. That's much more difficult for Kevin and others, to get to the de-risking. Same with BSEP. I've seen compounds that only had a BSEP, and there's, compounds in the clinic that have that. And so, but, I guess with the glass half full, right, we have made as much progress as we have understood. It is multifactorial. We have built the assays, and we know that certainly multifactorial toxicity with a low margin is a no-go. So, right, I think that's where we are today.
This is really a topic that will be dealt with in more details at our third and fourth seminar series. But essentially, it is going to talk about exactly this multi-parametric matrices that don't sort of do a weight of evidence approach of all of these different parameters that come together, and that you don't make a decision in any one of these assays. And important enough also to realize that they're also not independent from each other. There's a lot of crosstalk. There are a lot of compounds that might have reactive metabolism in mitochondria. Right, so the mitochondrial toxicity could be a response of that, or could be BSEP inhibition. There are ATP exchanges that happen in the cell between any of these mechanisms. So how do they impact each other? So there might be a sort of synergistic effect of compounds that hits 2 more than 3 parameters versus 1 might be more dangerous. An interesting question here from and you may not remember this, Kevin, without looking at the slides, but there was a question about case 1 and case 3 of your talk, right, where switching to pyrazanil over pyridazinil and pyrimidinol. So essentially the 6-membered ring with aromatic ring with different nitrogens in it in different positions. Has that been attempted? And we know that this is standard SAR, that people do that if the pharmacology is allowed.
No, and I think those would be good suggestions as a medicinal chemist, it's like, what's the next step? Or we know the pyrimidine in case study 1 was the culprit site. So how do we modify that ring, right? We contract it, make a different, 5-member ring, move nitrogens around. So those are both good suggestions on what would have blocked the bioactivation that occurred at the pyrimidine. Sometimes also remember that we're confined to some extent, not just on having a solution there, but we have to retain other things. and there are certain properties sometimes by introducing different types of the heterocycles that we lose the stability. We might lose, these were, in this example, CNS agents. So we had to get into the brain. So we had to have addressing PCP. So sometimes when some modifications become less viable from the chemotype standpoint to make it a drug, and sometimes we have to balance. So it's never fun to be a medicinal chemist because you have many things to kind of keep in play, right? And it's not just reactivity you're trying to optimize. But I think the suggestions in terms of proving the hypothesis, coming up with alternative solutions, by all means, they would serve us well. Right?
No, thanks, Kurt, for that question. I think that's the thinking of a medicinal chemist when he or she is going through the SAR thinking on this. A related question from Marcus on this is on the ketone. In case 33, do you find the ketone to be a problem?
We were concerned about that. That was when we were advancing forward that particular molecule, right? Before we got into aldehyde oxidase, there was a lot of concern about having a ketone functionality because they have a bad track record for toxicity. We actually didn't have any functional consequences to the ketone, and that's why it was still in play. And it ended up being that aldehyde oxidase became the major determinant for further optimization efforts. But that's very much in mind that the one thing that chemists oftentimes have, and we are very good at providing this, is structural alerts, certain substructures that are advisable to avoid. They have a bad checkered track record with bioactivation, thioesteins, furans, anilins, all right, ketones are another on that list. And so in that way, chemists have some idea on where not to take their chemistry to, because more than likely you will end up with reactivity.
A question from Rachel on the relationship between DILI and dose. And what are your thoughts on a dose sort of a dose as a mitigation strategy?
So what's challenging, we're not quite sure. We know that for intrinsic toxicants, right, there's a nice dose relationship, right? So acetaminophen is an excellent example. All of us, if we took enough tablets of acetaminophen, maybe it would be a little bit for me and a lot for Koshik, but we would ultimately experience the same liver injury. But the challenge is with idiosyncratic tox that some people can tolerate very high doses and have no consequence of this, where somebody who has those patient risk factors, right, at a much lower dose is very susceptible, right? So that's why with these idiosyncratic reactions, it doesn't have an apparent dose relationship within the population when we talk about migrating dose up. Now, dose we know is important, right? As I kind of illustrate from the GSK example, right? The higher dose drugs, the more likely, right? It's a more drug burden, right? Not just from a reactivity standpoint, but think about the mitochondria, greater exposures to disrupt the machinery of the liver, right? So if we can find ways to lower our doses, right? That's oftentimes, again, a focus in discovery, right? Optimizing potency, improving the pharmacokinetics. We place ourselves in a better position, right? Not necessarily to foolproof prevent reactions from occurring, but to minimize the likelihood. It's always about probability in the discovery space.
And the same applies to mitotox as well. I would think that is there a dose response for mitotox, Yvonne?
No. So sadly, as I said, it's of idiosyncratic nature, just because of the haplotypes and the mutation loads. So again, if you could do personalized toxicology, right, we just haven't done enough work on it. But for example, we did show at some point, if you take cell lines with different mitochondrial makeup, you do get different responses. Again, that translational piece, still needs to be worked out. So, I would hope, right? I would hope that at some point you can maybe do these type of things, right? Because again, if you would have known with troglitazone, what the susceptible patient population was, billions, right? Million people were happy with the drug. So we just, we have put a lot of that effort into personalized medicine. As you know, we do pre-screens, right, for certain medications for efficacy, but not for toxicology yet. So it's still.
Would you say though that compound drugs that are of low dose, let's say 10 milligrams or 50 milligrams, would be at a lower risk than one that's 400 milligrams?
That is no doubt, right? Yeah, is.
And is that same for activation as well, Kevin, that you would think that the odds are lower for compounds at low dose?
They would be. And I mean, you think about that covalent body burden slide that I have, right? That if you're already low dose and reactivity still occurs, it's still a very small amount of reactivity the body has to endure, right? More than likely, the body's able to process it. Acetaminophen, right, can happen where we can process that small degree of reactivity until it overwhelms the system or, unfortunately, alkylates the wrong protein. But low dose and reactivities, generally, it's tolerated well, right? So,
yeah, and I think Rachel.
we have to think about where are we with the dose discussion here.
And that's where I think Rachel was going with that there is perhaps the odds increase as you go up in dose, but even at a given dose, there is the idiosyncrasy of it. It still might not be predictive that you will or will not get it, but the odds get lower if the drug gets to a lower state.
And you can see this idiosyncrasy, actually. If you look at the ARIS database, you can see lactic acidosis for metformin. You can see lactic acidosis for linezolid, but it is not leading to death per se, right? And it's not hitting a percentage that would be concerning. But that is actually where you can see that mitochondrial component I spoke of, right? Same with the folks on silent Milas with the statin.
One last question for Kevin, and this is on the covalent inhibitors. And do drug-induced liver injury in covalent inhibitors? 30 seconds.
No problem. So excellent question. Covalent inhibitors, they will alkylate other proteins, right? We try and make them selective, right? These can be very safe drugs. We have many examples. We have our own in Janssen, right, with ibrutinib, right? So it's definitely possible to make safe covalent inhibitors. The intent is to minimize the reactivity of the warhead, right? So that it reacts just to the protein, but it's going to be challenging to hit just your target, right? How we often try and focus on them is also minimize other reactivity elsewhere on the molecule, right? The only insult should be coming from the warhead itself.
Thank you for watching this presentation. ACS Webinars is provided as a service by the American Chemical Society as your professional source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.